We'll be right back after this important word.
A joint. Ever had one? It really sets you up, especially at breakfast time. That lovely mild flavor. Gets you up quicker than anything, that does. Up and away. A good joint sends you off to a flying start. Puts you way out ahead of the rest of the crowd all day. Gives you energy to keep you going. Joints. Grab a basket and nip along to the grocer's.
Hey, welcome to episode 17, I believe it is, of the Operation Grow Show. Good to see everybody. Hope everybody's doing well. And yeah, it's the weekend once again. So it looks like we've got the usual crew of troublemakers. So we're going to have carry on the general discussions as we have been so far, going down the plant genetic side of things, and just generally see how things go from there. And I'll tell you what, hang on, I'm still pinned for everyone, I think. There we go. Yeah, it was just a hey, quick, how's everybody doing? Cascadian, good to see you again, sir. How are you? Have been up to anything interesting or exciting?
Planting the garden, man. Trying to get the garden planted. All the little babies are in their starter cells screaming at me, so I'm trying to get them in the soil where they can wiggle their feet and get back to happy, man. Yeah, that's
Nice,
what I've
nice,
been up to.
nice. Excellent, excellent. And Raptor, hey buddy, how you doing, sir? How's it going?
Hey, how's it going, everyone? I'm Raptor Grow. Good to see you all for another Saturday. Yeah, looking forward to learning with you all. And yeah, always a great presentation when Cascadian shows his knowledge and everything that he's acquired over the years. Yeah, should be a good show. Not much to say, just mushroom update. Spread out, reset my pot, my 65-gallon pot, shuffled everything around, gathered up all the mulch from the stalks and broke it down, re-amended everything before that, evened out the soil because it was kind of like topsy-turvy and uneven. And yeah, got some, mixed that up with some pink oyster spawn and been eating them since. So yeah, pretty stoked on it. But yeah, that's about it for the updates. Just gonna be eating here and doing some trimming while I'm listening. So yeah, looking forward to the show.
And how are you feeling after you had your nanobots installed? All good?
Yeah, pretty good. Just a little sore on the arm, a little groggy in the morning. Nothing some coffee and some food in my stomach can't fix.
Just wait till your superpowers come online.
Yeah. I'm just waiting for the feather tail to come in, and then I will be an actual good representation of a dinosaur, finally.
And you can hear every baby monitor for a 6-mile radius.
Yeah, super helpful.
Hey there, Shruby. I don't want to distract you while you're driving, buddy. How's it going? How you doing?
Not bad. Just heading home, like 2 minutes around the corner. So you safe then? The seeded crop got cut down last night.
Okay.
So driving right now.
Cool. So that's your first batch of seeds from the project. Yep. Nice.
200 to go through.
Sounds good. Well, I'll tell you what, you focus on getting home safely rather than worrying about the show for the moment.
Exactly.
Last thing we want to do is turn it into one of those reaction videos where it's our reaction like, ooh. Get to hear the paramedics go. Things you don't want to hear. GMF, buddy, how you doing, sir?
Oh, I'm doing great, guys, but I got to go. I just got a notification that Operation Grow started on YouTube, and I told them I'd be there, so I got to get going. Oh, wait a minute.
No, better get moving.
No, I'm good. Just, hanging out, doing my thing. It's raining here pretty heavy today, so no outdoor chores today, so a lot of indoor chores to catch up on. I figured I'd start the day and munch a ½ ounce of infused peanut butter chocolate ganache. So stuff I use for making truffles. I have some left over from a batch, so just going to start today with 15 grams of that and see where that goes. So that's about it. That's about all it's new and exciting. All right, we'll see. It'll be asleep or something.
Yeah, no, exactly. So yeah. Yeah, generally things are going well, I think. Looks like, yeah. Well, at least in the Northern Hemisphere, summer's coming. I spoke to my brother today, he's in Saigon, and he's like, man, it's ridiculously hot. It's like 40 degrees Celsius and 100% humidity. So the walls are just dripping. He was talking about how they get it really cold down to 18 degrees Celsius with they all huddle in one room and just crank the AC up. And I go, huh, it's currently 14 degrees. I'm walking around in a t-shirt and flip-flops outside. How relative is shit, I tell you what.
So they need a really big dehumidifier for the entire, and that's a big dehumidifier they need.
Yeah, exactly. So yeah. I should stop saying all the bloody time. Yeah, Cascadian. I don't know. I feel like unprepared myself on this one.
Oh, this is gonna be fun then. So am I. No, I'm kidding. So yeah, today's let's see here if I get the screen share going today.
Let me enable that. There we go.
Today is a continuation of last time. I'm sorry right now. Here I go with the ums. All right. So we're back to the basics, part 2. I'm gonna fill in a little bit more gaps. I'm gonna hurt your head again. This is another talk of probably 6 or 8 weeks of lectures I received that I'm getting ready to jam into about 20 minutes, guys. So good luck. You're probably gonna have to watch this a few times. I'm probably gonna glaze over a couple of things, not include all the nitty-gritty details because we would be here for hours. But today we're talking DNA and RNA, transcription and translation, introns and exons and telomeres. So it's all some of the details behind how DNA and RNA and how you get from genetics to proteins. And then later on, we'll be able to understand how, in further talks, we'll be able to go into how things are mutated, how things are changed, and what that does to the plant, how that actually means something to us. So this first slide is just a comparison of DNA and RNA. You can see that they're made of some of the same nucleotides, but at the bottom, you'll see DNA contains thymine and RNA contains uracil. If you look at the structures, you'll see it's fairly similar. There's only a couple of small changes between thymine and uracil, but RNA communicates using uracil and DNA communicates with thymine. So it's all the deeper I want to get into that right now, but it's important to understand that they do use different alphabets to talk. So DNA is in the nucleus. That's where the genetic code is stored, like I was saying before. And this is a very high security place. And RNA is the messenger allowed to go into the nucleus and come out of the nucleus with this script of information. So there's a replication process when DNA makes more of itself, which is usually in a cell replication sort of sense. And then parallel to that, there is transcription and translation, which I'll talk about in a second. So this is kind of one small branch of how DNA works, and then we're going to travel a little bit farther down a secondary branch of why this is all important. There's a lot of really big words on the screen right now, and I don't want to get into all of the details, but I want to, oh, hey, I didn't change that. Ignore the, ignore the text at the top. That's from the previous slide. I should have taken that out, but you know, we are all human. We make mistakes. So
I did just want to ask one quick question before going. Just
Yeah.
just to be and just to make it put it maybe into context a bit more here. When we're talking about RNA going into the nucleus and going through and coming out, what is the purpose? Why is the RNA going in? What is it up to? What is the reason for it doing that? I mean, we talk about the replication of DNA, but what is the purpose for? Yeah, it's dipping in and it's reading code and disappearing out again.
Yeah. So let me hold you off for just a minute here. Let me get through this replication part, and then I'll dive deep down the rabbit hole of RNA. This particular step is DNA making more DNA. So there is a set of RNA and DNA polymerases. The RNA polymerase I'm going to talk about in a minute. DNA polymerase is what's doing this, and this is a conjunction of different molecules that come in and make this happen. So I think there's 3 polymerases. You'll see the one at the top here is pol alpha, and then the one down here is pol, is that a beta? I think that's a beta. So they're different components. They're both polymerases, and they're all involved in this replication process. DNA is replicated from the 5 end to the 3 end, which has to do with the carbon anyway, too much detail, but it's replicated from the 5 to the 3. So that's why this bottom part is a leading strand, is because it's actually writing on the 5 strand as it goes. It's actually writing the sentence in order. Above that, it's called the lagging strand, and it's writing backwards on the 3, from the 3 to the 5. So it's writing backwards. So it has to write a chunk of it and then stop and join that chunk to the part that it's already written, which is what the DNA ligase is doing. I'm pretty sure. So it'll write a section and then it'll join it, and then it'll write a section and then it'll join it, because it's writing it backwards. So when DNA is replicated, part of it is written as, the cat crossed the street, and the other part of it is written as if I said the cat crossed the street backwards. I'm not going to try, but take all the letters, flip them, and you have one written correctly, one written backwards, and then it has to take the backwards ones and put them in order so that it all makes sense. You can see how on the lagging strand, when it's trying to put things back in order, how mistakes can occur, and that can lead to funny things in gene expression down the road. So that's why I wanted to talk about this, is because the mechanisms can actually make mistakes. Those mistakes will translate into differences down the road. Whoa, did my screen go funny for you guys? Nope, there we go. How do I get that to go away? There we go. Okay, so that was DNA making more DNA. This is transcription, and then translation is DNA turning into a protein. So it's not making more DNA for the purpose of cellular division. It's taking the code and going to a protein to then go build something in the plant. So Pete had a question a second ago, and now I'm blanking on it. It was something down the road of why this is important. Can you repeat your question, Pete?
Yes, I just realized I was talking to mute. It was to do with, putting into context, you've got this RNA coming into the nucleus, going along a strand of DNA, and then whipping out again with an instruction set or something taking with it. So what was, apart from, the reason why you've got these very specific bits of RNA that dip and dive in and grab what they need to and dip out again. But this is, I guess, what you're alluding to here.
Yeah, so RNA has like the security code to be let into the nucleus, because the nucleus is very particular about protecting DNA and keeping it safe, because anything goes wrong in the DNA, and it has vast ramifications across the whole organism. So RNA has a special tag that says "Let me in," and it has the ability to get into the nucleus. Other molecules and things are so small they can slip in, and it gets kind of fuzzy, gets kind of dangerous. But the nucleus overall is very picky, very, very finicky about who it lets in. RNA has the ability to get inside. It takes these RNA polymerases now instead of DNA polymerases, and it unzips the DNA and reads from the 5 to the 3 again, and turns out mRNA, I do believe it goes from it comes in with an empty piece of paper per se, talks to the DNA, and the DNA tells it what to write down on the piece of paper. It takes its piece of paper. So that process of writing down on the piece of paper what the DNA is saying is transcription. That's what's being alluded to here on the screen. There are 3 parts of transcription. I wanted to show this so that you could look into this deeper. Those are the keywords to look up, but I don't want to spend an hour breaking it down. But I did want to show you guys that these are the 3 terms. These are the 3 steps of transcription. This is the overall how it works. It unzips it, it reads it, it zips it back up, and then you have this RNA strand with this piece of instructions on it. So then the RNA leaves. Come on, computer. There we go. The RNA leaves the nucleus, goes into this funny little thing on the side here called the endoplasmic reticulum, and attached to the rough endoplasmic reticulum are ribosomes. And the mRNA goes to the ribosomes, and ribosomes are made of 2 pieces, the top portion and the bottom portion. They have names, but I'm just trying to give you guys the idea, not drown you in vocab this week. So those 2 pieces come and they clamp on the mRNA, and they start to read it. There's a particular order of nucleotides, and reading that on the mRNA, using uracil, not thymine, so that's what the U's are here for. It then translates, which is why it's called translation, that code into codons, these three letter sets that then specify a particular amino acid. So as this code goes through the ribosome, it codes for these particular sets that lead to particular amino acids that are then joined together through peptide bonds to make polypeptides. Each one of these amino acids is also a peptide, polypeptide, many peptides stuck together with peptide bonds. So then you end up with a protein, essentially. So that's taking information from the nucleus, from the DNA, where everything important is stored, and getting it to something that the cell can then spit out into and actually make more pieces of itself. Like it's building itself here. So that's a rough introduction to transcription and translation. It happens in the nucleus, goes to the endoplasmic reticulum, and then the endoplasmic reticulum sends it to more anatomical pieces of a cell before it's packaged into a vesicle and shipped out. This is all pretty important because mistakes can happen in these steps that change the protein structure. And when you change the protein structure, it changes what the protein does. It has a structure function relationship. So mistakes in here will code for proper or improper proteins that lead to proper function or improper function based on mutation or mistake or coding errors. There's a handful of things that can go wrong and throw wrenches in the system here.
And that's
So
just it, because if there's the bug in the system being a coder, the bug is in the DNA, so that when it gets broken down and stripped out into the strand of mRNA, when a certain circumstance arises that suddenly results in the body needing to produce this, as an example, or whatever, the organism to produce something. So now at this point is the first time it gets tripped up. So like a stress response that suddenly results in a mutation expressing itself because it's the first time it's been called upon to be produced, and it makes that error mistake. So not all mutations are deadly, for example, or at least you could have them and they're hidden until you trigger an environment situation that causes them to suddenly come up. But yeah, I mean, and this is really complicated. I mean, I saw a great example where somebody went, okay, here's this a gene, well, just a small section of it, and it was just a row of letters, and it go, okay, put you in front of a typewriter, and you get one go at copying all of this down letter by letter, and you get one go at it, and tell me you're not going to make a mistake somewhere along the line, there.
Right.
And as you say, if it's the right kind of mistake, and affect protein in a certain way, that can be pretty dramatic at the end of the day.
So this, while you were talking, it occurred to me that this is the process that takes place between the genotype and the phenotype. When you alter the environment, the plant has to respond, and the plant goes back to the DNA and says, okay, we were using this set of code, but now we're in a place where we need this other set of code. And it will, in layman's terms, it'll essentially shut down the first set of code and activate the second set of code. And then you have the RNA not reading the original set and coming to read the new set, synthesize new proteins, which then changes the phenotype, which makes the plant look different. Mistakes in those processes then lead to irreversible mutations or changes in the phenotype. Because you've changed the genotype, that's where the mistake is. Even if you change the environment again, that mistake goes with you because it was a genotype error, not a phenotype difference. So that puts a little bit more context on kind of why these particular steps are important, is because this is the coding in between the genotype and the phenotype that you always hear people talk about. This is what's actually happening. Let's see what the next slide is. Kind of a guess for me too. Okay. Again, ignore my text up top. I forgot to change my text boxes. Introns and exons are portions of the DNA strand that either contain the code that they want to read, or they contain a sort of buffer between, like it's like having a word, having a space, and then having another word. The intron is the space between two words. The exons are the words. So you can see here how the exons translate from DNA to RNA to mRNA, and then the mRNA is read as a solid sentence to create a protein. It takes all the spaces out, so it's just all the letters of the sentence in order, no spaces. And that's how the protein comes to be. But it's important to understand that there's introns and exons, and that there are these particular gaps or spaces between the words that not all of the DNA from front to back is. It's all important, but it's not all read and translated to proteins. There are buffer spots because mistakes are made. There's these buffers to separate things so that they don't accidentally code, say, the back half of this blue exon into this whole green exon when it wanted this whole blue exon instead, but it started reading at the wrong spot. it's going to read for a certain length, but it has to start at the right spot, which is the end of the intron, the beginning of the exon. But if it was to pick the wrong spot to read, you could see how if the exons were together, it would read the middle of one exon to the middle of the other exon, and you would get something non-functional. So I wanted to
I
get
know a
people
few
in there.
people where that's clearly happened.
So
More
yeah,
than once.
it's important to understand that these exist. How much detail you need depends on what you're trying to do with this plant or in life. So this is really just an introduction. I'm giving you keywords to either quiz me about or dive deeper yourself, depending on the type of individual you are. I'm totally open to questions, interactions. I can't see chat, so I'm sure there'll be some questions when we get done here. But that's some of the intricacy in DNA, RNA, and this whole process. I'm just giving you guys tidbits to try to fill in some of the things I didn't have time to give you last time. And then, telomeres here. Telomeres I mentioned last time. Whoa, buddy, back up. My computer's freaking out. All right. So telomeres are at the ends of the chromosomes, if you remember from last time. And this picture just took the words out of my mouth, basically. This is a progression of cell division. And you can see how the telomere length on the original cell is quite long. And every time that cell divides, those telomeres get shorter. Part of that is shaved off in the process. And this is believed to be part of the mechanism behind aging, behind some of the things that people suffer from due to living longer. It's because these telomeres get shortened and eventually they disappear when cells divide enough, and the replication process, cell division process, actually starts to shave away important information on the ends of the blue part, the important information on the chromosome, because it ran out of the yellow telomere. So every time cells are divided, you're losing a bit of genetic information it's like replicated code on purpose that doesn't have a function other than to get shaved away. And as it gets shaved away, you age, essentially. And then you get to a point where you start to break the chromosomes because you're shaving away important information. So that's just another tidbit I wanted to throw in because we didn't have a time to really get to it last time.
Do you know, in the term, when it comes to doing something like tissue culture, does that reset telomeres?
I don't know. That's a fascinating question that I don't have the answer to.
And one of the things about telomeres in cannabis in particular is that cannabis has particularly long telomeres, which means that's why you get these cuts that get kept and can keep going and going and going. But eventually, I mean, if they go for long enough, I mean, theoretically, they eventually start to freak out.
Right. Yeah, I need to look deeper into sort of telomere regeneration in plants. I'm not sure if it's possible. I'm curious if there's mechanisms behind it. But yeah, that would be fascinating to know in the context of tissue culture, how telomeres are affected, for sure. I need to look into that. So maybe we'll have time in a further week to get into that.
The only way that we might know of for now is just putting it in stasis of a sort. So just slowing it down until it's just a little piece of genetic information, letting it grow out. Or that combined with cryopreservation. So yeah, just some interesting
I have read studies done on telomere degradation, but it specifically is typically in chimps. And from the reading that I did on the subject, what they found was it literally has to do with your metabolic rates and how quickly you are using up those telomeres available. So for example, when they took chimps and they cut their calorie intake by 33% compared to the other control group, they ended up living longer as a group simply, and their telomeres ended up being shortened at a slower rate simply because they were going through fewer metabolic processes of always digesting, et cetera, et cetera. So they were doing less cell growth. And so literally had a longer lifespan from not tearing down those telomeres by constantly replicating. But that's a study in chimps. I would assume the mechanism has got to be somewhat similar in plants, not knowing whether tissue culture could reset it. But, you know, especially in that context, they found a reduction in calories can lead to a drastically extended use of those telomeres.
That's interesting. And that makes sense, you know, why a healthy diet could lead to a longer life. You know, you eat the right food, you don't need as much of it. You change
People
your
don't eat
metabolic
as
rate.
much.
Right.
People who maybe eat only, you know, limited diets on certain times a day. I mean, there's a reason some of these aesthetics were able to live
Yeah.
long lifespans, and that could have something to do with it. We don't know, but that's the earliest information we have from chimpanzees anyway, on telomere degradation. And my personal hobby, part of the reason for the handle I use is just my fascination with epigenetics and how they seem to influence, you know, human interaction, human behavior, and our own lifespans. You guys have heard me gone off a couple times about, you know, the effects of World War II and the Civil War on actual, you know, genetic markers that we can watch. So I don't know, it's a fascinating topic. Thank you for taking the time to get into it, Cascadian. I really appreciate you edifying us all on, you know, the nature of RNA and DNA, because it's something a lot of us learned in high school but don't remember or don't use.
Yeah, I seen some when I was preparing the slideshow. I seen some topics talking about stress induced, how stress affects this. I kind of glazed over it all because I was just trying to get these basics down for us here today. But I definitely seen how, like you were alluding to, how war or stress or conflict can lead to the degradation, essentially, on a genetic level. So this last slide, well, second to last slide, basically the last slide is just a rehash of what you seen last time to kind of keep things in context. I really have done a better job this time of telling you why it's important while I told you the information, so I don't have a big spiel to give for this. But you can see on the left an actual pretty good representation of a DNA structure, and then all the terminology from last time and the bonds and the I noticed now I didn't include purines and pyrimidines, so I'll have to do that next time. And that the reason, I guess I could give you a little bit here right now, but the reason it's important is because they have different amounts of bonds based on who is talking across the DNA, which leads to different structural differences. You can see over here on the left, there's a major groove and a minor groove. Those are indicated basically by the shape of the DNA, which is partially influenced by the amount of bonds between the nucleosides. So I'll try to give you guys a little bit better breakdown on that on next time. And then we can start getting into actual cellular replication and how a chromosomal crossover refers to a process that happens during cellular replication that changes the layout of the chromosomes. So we'll start to get into things that are actually a little bit more interesting instead of all these details as far as how it applies to us trying to grow these plants and breed these plants. So with that, thanks for putting up with me again. It's always fun to get on here and present.
No, thank you. That was great. That was really good. That was a really good presentation. It's a complex topic, and I think that was covered. Yeah, that was covered very well. I'm just trying to see if we've got general feedback from chat. Is that it's been very interesting. I just want to get an idea of any questions coming through there. I mean, I know it was a bit tongue in cheek. Well, I know it was tongue in cheek of Jade. This is one about mRNA can alter DNA, but actually, that's the very reason is that it can't. It can read what's there, but it can't go in and make a change to what's there. And it's probably part of the security behind it having a slight. It has the T instead of the. Well, it has the U instead of the T, which again just allows the body to distinguish. Okay, you're not DNA, so we're not going to pay that kind of attention to you. You're not a random loose strand of DNA floating around.
I really thought the earlier side conversation on telomeres was interesting, and especially what GMF brought up with reducing calorie intake and the effects of shortening telomeres, or at least extending life. Because think about what the plants had to go through over millions of years. Now, there was so many periods of them just going through with the bare minimum. So of course they would the genetics would be adapted to that, instead of just this overbarging of all these nutrients and everything like that. I'm sure that it can handle it, because if you find an abundance of nutrients, you're going to take advantage of it. Whatever life form it is, a mammal, reptilian, amphibian. But yeah, just got my gears turning. Thought that was cool. Thank you.
I mean, it would be interesting to see if you could get to the point where, okay, now I know what I could foliar feed, spray my plants with the correct mRNA sequence to tell it to produce the protein to make purple, to make this specific, so not genetic modification, but just knowing how to play the game, knowing how to signal it, telling it, okay, I know what your instruction set is, so I can just feed you the correct instruction set on demand. I don't have to edit the actual, I can just bombard you with this and you'll pick it up and play with it. If that makes sense.
Well, and I mean, just, in a very practical sense, remember what that M stands for in mRNA. People like to use acronyms, but the M is probably the most important part of that whole thing, because it stands for messenger, right? That's all it is a messenger.
Yeah, which is the act of going from the nucleus to the endoplasmic reticulum. That's the messenger aspect for sure. Is it travels back and forth. Don't shoot the messenger kind of thing.
So if you can get in outside of the nucleus, sorry, Jimmy. So yes, if you're introducing this outside of the nucleus, you haven't had to worry about hijacking that security system. You're now just sending the messages directly to where you want them to go. And you're being very precise about what messages you want sent.
Well,
that'll be in the next nutrient line. Comes with mRNA to make big birds.
Well, I really see the future of breeding as we advance more and more. All this is obviously necessary. Like, we're gonna, just like you were saying before, we can insert genetic codes to perform certain actions. Like, look at what we've done with yeast. I believe it was a couple of decades ago. Forgive me for not remembering the exact date. But yeah, yeast, like, we used to get insulin from cows, from the organs of cows. Now we have yeast that produce insulin. So it's going to be interesting how that's going to play out, but also how we can use all this information to breed more responsibly, so we know exactly what we're breeding with. So we won't necessarily need to insert anything. We could just let it go in, take our time, but this is a much more exact way of breeding. It's not going to take decades to get where we want to be. It'll take, a reasonable few years or so. But yeah, just from observing and taking notes of like what kind of genetics you're dealing with and how to integrate that with your goals. Yeah, but I'm very happy to hear all these explanations that Cascadian's been presenting to us, just because these things need to be made clear. No more bro science. It needs to just be put out like, this is what it is. You can't talk your way out of this explanation. It's proven. So yeah, it's I am looking forward to just like the bullshit kind of going away. But yeah, also what people call bro science, some of it actually is valid. It's just not done in a way that it's not recorded properly. It's not proven again and again in the right settings. Like it has to be repeated again in exact same settings, and then slowly change up the variables from there. So yeah, I have no problem with people discovering things. It's just you have to kind of prove it more than 1 or 2 times. You're gonna have to prove it a couple million at least. So yeah, I'm very happy to hear all of this this information going about, just so I can stop explaining it.
I'll jump in here too. Like there's, and this is maybe a little antagonistic of me, but I'll go ahead and do it. The truth is, people try to bring whatever they want to science, right? Like someone in chat was just mentioning, and I won't call them out by name, funny how this all came together randomly, trying to be all tongue in cheek. Well, the truth is that's not random. This entire system has been built over a long time, and it's gone one step at a time. And all these different things we're talking about in genetic replication, they're not mistakes, they're experiments. And you look for ones that actually excel in the environment, and that ends up becoming speciation. Like it ends up becoming its own genetic dominance. So it's not random at all, but it's also not planned. It's a progressing series of experiments. It's growth one on top of the other. That's how we get, simple organisms all the way up to complex organisms. That's how we've got, 4.5 billion years of microorganisms on this planet, all the way leading up to us, a complex organism. So
yeah, exactly. And we're only one step on that path. I mean, that's the other bit that's really interesting. I mean, yeah.
In another one snapshot of a long story.
To 500,000 years' time will be completely different. We'll be looking back at this lot going, oh, God, look at that lot. Look at that lot.
I saw
only had 2 arms. I mean, Christ. How did you drive and jerk off at the same time?
Right? I actually saw a comment in chat. It's very succinct. But science doesn't disprove anything. Science science doesn't prove anything. It only disproves. Like
it's an answer to the question how, not an answer to the question why. And that's a big difference.
There was I'm blanking really hard right now on the details, but I had my biology professor tell my whole class about an experiment that was done where they basically recreated the atmosphere before the rise of oxygen levels, and they took for lack of a better term right now, they took a genetic soup and they put it under the atmospheric conditions of the atmosphere before it became highly oxygenated, and they just left it and watched what happened. And they found that lipophilic, the membrane of a cell just organized itself naturally. It just naturally started to create. Now, it didn't inherently make a circle, but it made these strands of I'm blanking on the word right now. What's it called?
Amino acids. I mean, they made the basic building block of life is what they essentially produced.
They found amino acid structures. They found the cell membrane structures. They started to see all of these different pieces that we see in the more basic side of cellular structure start to just organize themselves in this soup. And it just happens. Some of these chemical reactions and these attractions just are in place when those things all come together. It's fascinating. It's fascinating because you start to get, you start to feather that line of, I won't say the words, but it's really cool how it just happens naturally. It just occurs. And then from that becomes more organization that leads to cellular structures, cells, and then you start getting to bigger organisms, molecules, and here we are. Yeah,
if anyone wants to look this up, please feel free. My science teacher in high school was big on this. The guy who did this research, his name was Sidney W. Fox. He did a lot of the research actually at Southern Illinois University. I think he was, if I remember correctly, he made something called proteinoids, which he was able to create amino acids that would function in the same way that our DNA and RNA do, but they're not nearly as complex. So this series of experiments Sidney Fox did was able to actually create this in a lab atmosphere. He worked at University of California. I know he worked at the California Institute of Technology. Like he was free, if I remember correctly too. I think he worked under another famous, was Jenner or someone like that. No, it was later. It was a different science. But anyway, you look up Linus Pauling. That's who it was. He worked under Linus Pauling. So yeah, look up Sidney Fox, and you will find some very interesting work that he did specifically around this topic. And it was in the 70s. It was from like the 50s to the 70s that he was doing this work. Sorry, I'm spastic, but I like that guy a lot. Sidney Fox is a cool dude to read about. Yeah, he taught at the plant biology department, I think, at Southern Illinois University.
Yeah. And thing is, this is the breeding project. How's everybody's projects coming along? GMF, how's yours? How things going there with yours? If you don't mind me asking.
Oh yeah, so far excellent. I can't really complain. Again, just keeping my typically it's that deep chunk male, and I've been just kind of looking to see if I can grab that trait that it has to throw roots out of literally any branch. Like sometimes 3-4 feet up on the plant, it'll throw roots off a branch, and I'm really looking to see if I can pick that up and get that to transfer without taking over other things. So right now it's pretty interesting. I'm watching the morphology on some small ones that I've crossed with the deep chunk just to see how much deep chunk is going to step on things, because the rumor is always that it does quite a bit. But yeah, so far I'm seeing some very different morphology with things that are kind of, so I don't know, it's pretty interesting. I'm enjoying it, but again, I got many generations yet to go before I'll be able to hopefully pick that trait up and stabilize it in something that doesn't look like a photocopy of deep chunk.
Nice. And Shribi, you were saying as you were bumming along in the car, how are things going with yours there, buddy?
I chopped them all down last night. So I'm waiting for the first batch of seeds to dry out. And I'll start popping some more. I'm going to focus on the AK cross first. Out of the 3 AKs I popped, there was one keeper I found, and the Storm Shadow male. It's very promising. I'm trying to increase the bud structure of the AK while maintaining its coloring and hopefully a little bit of its aroma. But as in the terpene profile, I'd prefer to keep the Storm Shadows if it could.
Nice. So you're going to have a good deal of seed chucking coming up.
Oh, fuck yeah. If only
how do you like to do that, can I ask? Yeah, right? Are you planning to do it manually, or are you going to try to, like, what is your plan, bud?
It's not a big plan, so I plan to do it by hand. I'm thinking at most it's going to be like 100, 150 seeds. So shouldn't be too bad.
No, that's not bad. When you get up in the thousands, you're talking a longer day by hand.
But yeah, I plan just to dry it out as a trim bed.
Hey, we happen to know someone. Oh
Shit must
yeah,
go.
man. I tell you what, that would be. Yeah. Oh yeah. Just doing it for a few hundred. I remember back in the day. Um, was the case. And yourself, there, Raptor? How things going on your front?
Pretty good. I just chopped down the Scarlet Begonias. I really like um, the two different ways they express themselves. One was shorter, one was taller. The shorter one had a green apple champagne taste to it, flavor. And the taller one had a green apple candy, so like Jolly Rancher or yeah. But once it's all chopped down. A little piece fell off on the side. I don't know how it happened. But yeah, I'll take advantage of that fact and put it in a bowl, and that actually tasted like a green apple taffy. So again, going back to the whole green apple candy flavor, I need to look up that specific. I don't even know what I would call it at that point. I'm sure GMF could enlighten me. But yeah, I do. I really like what I got out of it. I only popped 6 seeds, and I think I'll have to talk to Jeff into ordering another pack. It's not on the market anymore, so I'm just gonna have to work my charm, see what I can do. I'm more than happy to pay for it. The actual genetics were a gift. I won it on Embracing Organics. Thank you to Embracing Organics and Irie Genetics for the gift, the prize, I should say. And yeah, I'm definitely interested in it. What did happen is that I left a Scarlet Begonias male in there, and it was super short. And I was just trying to extend the life so I could keep taking cuts off of it. At the time, I didn't have the space to put it in another tent. I only had one tent. Got to make do with what you have. So I made sure to check everything before I left on a trip, a couple months ago. And right when I came back, I got lots of pretty flowers coming out. So I left the exhaust going. So it was a short flowering plant, and it was a short stature as well. So I'm interested to see it. It totally bombarded both of them, both the girls. So yeah, definitely hearing tink, 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 tink as I'm doing some trimming. So yeah, I wouldn't necessarily call that breeding. I will see, what those express. And if I see anything favorable out of that, I will save it and put it to the side and explore it some more. But in no way is that the proper way to go about this. As we've gone over in the past, there's certain steps that you might want to look into. And it also has to do with what are your goals, as I've been learning more and more. So I'll keep that around. But like I said, I'm looking into getting another pack so that I will run with no try to do it as perfect as possible. There's a lot of variables that happened during this last run that made it, while I did get to learn a lot about the plant, I didn't get to learn about it in its true state in a responsible manner. So yeah, I'm looking forward to that and telling you guys what it brings in the future.
Sounds good, man. Nice. And your cascading, you were having was it the root beer that you were playing around with there?
Yeah, that all got whacked down and reset because I had well-documented Hermie issues. No surprise. So I'm currently getting ready to do a different cross. I got the Blueberry Blast lady from Snow High, and then I got Blue Dream Grape Soda Skunk from a friend. So I'm getting ready to make a sort of a Blue Dream mashup. Blueberry Blast and the Blue Dream Grape Soda Skunk both have Blue Dream for moms. So when I make the cross, grandma on both sides will be Blue Dream. Grandpa will be Johnny Blaze, and grape soda skunk. So it's a blueberry haze, a blueberry haze, a haze blueberry, and then the polyhybrid that is grape soda skunk all coming together. So I'm pretty excited about that one. It should be very much, should be a lot of vigor, should be a lot of baseball bat type colas, should be very much a commercial appealing sort of combination of genetics. So I kind of want to take that project and steer it for commercial cultivation for 3-foot baseball bats of beautiful blueberry haze flavors. Yeah, so that I'm really excited about that one now. I've got to reset my whole coffee, chocolate, caramel, mocha project. It's getting a little bit more complex, but it should turn out to be a cluster of genetics as opposed to a single line. So I should be able to create a handful of interconnected or slightly related lines depending on how I want to do it. Yeah, so I'm diving off into blueberry haze world for a minute while I try to restructure the coffee, mocha, chocolate, caramel stuff. But it's all exciting.
No, that's sounding like good fun, man. All right, chat's kicking off. Great fun. Good laugh. Chatter. It's always good to see different opinions being expressed.
It is, though, because that leads to critical thinking, and as long as nobody gets their feelings hurt, it's all for the best, because it encourages that, you know, what you do support has support.
Yeah.
It makes everybody think. It's a good thing.
Yeah, that's again. Yeah, I can only speak for myself and speak personally. I'm from the world of science and like to go from that side and see how things are done, and needs to be observable, repeatable, predictable. To me, that's where the explanation comes in. No, but that's
It's not
again, that's just me.
repeatable. You didn't disprove anything.
But hey, that's half the fun of what we're doing when we're trying to cross things, get things to work. People say, "Oh, that can't be done." Well, hell, let's give it a go. Let's see if it can be done. I mean, that's how it should be approached. If somebody says, "No, that's not how it is," well, okay, well then fine. Show me that if that is. If you feel strongly enough about it, then either go out and prove it, do the cross that everybody said couldn't be done. Find the information. But yeah. No, that's good. And I see what that brings us almost pretty much up to the hour. And so on that note, if you would like to jump in and join us for the after show chat and hang out for a bit, link is going up in chat now. So there we go. You're more than welcome to dive in and jump in for a bit, and we'll get to chat for a little bit longer. But thanks again, Cascadian. Now, next week, it may not be a show. I'll have to see. I've got to go and do an installation up in Oxford at a greenhouse there. So I may or may not be able to make it back in time for that. So might just have to play it by ear. There may be a show. There may not be a show on next week. We'll just see how it goes. But yeah, thank you very much, Cascadian, for another great presentation. What I'd like to do maybe for the next one after this will be do a follow-up on the environmental one and just take a look at, like, as I said, things that produce heat in the environment, like, and how they produce heat, the different ways they do it, and then also to take a look at, what produces humidity, and then maybe different ways of controlling it and the like. So yeah. Where can everybody find you, Cascadian? Just on that one.
Yeah, Cascadian Grown on Instagram is the best place to find me. If you have any questions about today's lecture or any of my previous talks, please feel free to DM me. I'm completely open to questions. So yeah, look for me there. You'll find me here whenever we're live again. I'll be here too.
Excellent. And Shreeby, where can everybody find you, sir?
You can find me on Instagram, ShroobyDoo. And on Cannabuzz, the Growing Buds.
And also, you've got your show on, is it Mondays or Tuesdays? Mondays.
It's on
isn't it?
Tuesdays.
Tuesdays.
The Grow for Show.
That's the Grow for Show one, yes. And then there's also the neighborhood. Oh God, there's
Grows
so
on
many. I
Mondays.
can't, that's the one. Neighborhood Grows on Monday. Yeah, yeah,
Yep.
yeah.
Same time.
No. Same time, same place. And GMF, where can we all find you, sir?
Oh, sorry, you can typically find me hanging out here with y'all on Saturdays or yesterday at Grow Room 420 on Friday. I'm trying to live on a daytime schedule, so the nighttime shows are really hard for me to make now just because I'm trying to be a responsible human being. But so it makes it a lot harder to be up for the late night shows. But if anybody's got questions about anything, if you just want to have a discussion, if you got food questions, you want to know how to make something, hit me up on IG, Genetic Memory Farms, and you'll find me hanging out in the neighborhood on Monday. We're always there. Invite you all to come by. We're pretty much open to whatever. So yeah, it's thanks, Pete, for making this platform for us, I should say. And thank you, Cascadian, for, how shall I say, edifying us on the topics of which you are a general, big, smart guy with a big brain. So thank you very much, man. I appreciate it.
Hey, and Raptor, where can we find you, sir?
Hello, everyone. Very informative show. I enjoyed it very much. I am Raptor Grow. You can find me at Raptor Grow. Hopefully everyone can see here. I'm not just pointing to the ether. But yeah. Please join me and all my friends at the Neighborhood Grow Show at 4 p.m. Pacific Standard, 7 p.m. Eastern, 12 a.m. UK. Sorry for the UK. If you're up that late, you're more than welcome to join in, and don't feel like you have to watch the whole thing. Rest. You can always catch up later. We record it and we post it almost immediately. So yeah, please. You can also find me at Operation Grow, same day, time, 4 p.m. Eastern and 9 p.m. UK. And I feel like I'm missing something. Oh yeah, GR420 Forum Grow Room. So
yeah,
they do great work. You can basically see me there for an hour, a couple hours with that lot. Really excited to just branch out into European, like different European groups of cannabis growing. So started off with Pete. Thank you so much for having me. It's been a lot of fun and I've very much enjoyed it, and I look forward to the future and how we grow.
Cool. Excellent.
No
and
pun
you
intended, how we grow.
And you can catch me at the Operation Grow 2019 on Instagram, as well as Peel Grow Systems on Instagram. You can get to the website. Dear old Remo's been posting links to it. Thanks everybody there. But peelgrow.systems. And also just a big thank you to, I know I don't really mention it very often, I think I've barely mentioned all actually, but a big thank you to all the patrons for their support and subscriptions. It definitely helps cover some of the costs for doing the show, so thank you very much. And yeah, once again, if you'd like to dive into the after show, you're more than welcome to. But on that note, we may catch you next week, otherwise we'll be here the week after. Stay safe, stay well, and thank you very much for being there.